Hi everyone, welcome to my channel Pure Biology, and today I come with another topic related to cell biology, that is the plasma membrane, the structure of the plasma membrane. Okay. Before continuing this topic, I want to say thank you to all of you who have already subscribed my channel, and I request you, if this channel is helpful towards you, then please you support me by subscribing my channel and sharing my channel. Okay. And today's topic, that is the structure of plasma membrane. Okay, what is the plasma membrane? Plasma membrane, actually this is the outermost covering of the cell. Okay, in case of plant cell, the cell membrane is the outermost covering, but in case of animal cell, this cell membrane is the outermost structure of the cell. But in case of plant cell, there is another structure present just outside the cell membrane, that is the cell wall. Okay, so here first, this is the plasma membrane. It is the fluid structure. It is the fluid structure and forms the external boundary of the cell. I just have told you that it is the external boundary of the cell. In 1972, Jonathan Singer and Gerd Nicholson. Okay, Singer and Nicholson, those two scientists who proposed the fluid mosaic model. That means they proposed the structure of the plasma membrane is just like the fluid mosaic model. Okay, and in this model, membranes are viewed as fluid structure, quasi-fluid structures in which proteins are inserted into the lipid bilayers. Okay, in which the proteins are inserted into lipid bilayers. Next one is that chemical constituents. What are present in this plasma membrane? First of all, lipid is present. Bilayer structure of the lipid is seen here, and also present protein that obviously embedded within the bilayer structure of the lipid, and carbohydrate also present. Okay, but here the ratio of the protein and lipid that varies, that depending on the cell types. According to the cell types, the ratio of the lipid and the protein that may vary. Okay, but the carbohydrate that is present 5 to 10% of membrane mass. Next part is that lipid bilayer. So here this is one unit structure of the lipid, just a very rough structure has drawn here. That is the hydrophilic head and the hydrophobic tail. Okay, hydrophilic head means this likes water. Okay, and hydrophobic tail, that means they remain or they try to remain just opposite side of the water. Okay, so this is very rough structure of unit lipid. Next, lipid bilayer, the basic structure of the plasma membrane, that is the lipid bilayer, and this bilayer is composed of two leaflets of amphipathic lipid molecules, and whose polar head groups are in contact with intra or extracellular aqueous phase, whereas they are non-polar tails that face each other, constituting the hydrophobic interior of the membrane. What is saying that? That there are two parts. One is the head part, another is the tail part. And the head part, that is the polar portion, and the tail part, that is the non-polar portion. Okay, so head groups, they remain contact like that. Okay, they remain in contact and with intra or extracellular phase. Okay, they remain contact just like this. These are the tails and these are the head. Okay, so this is the head and This is the tail. Okay. In that way they are arranged. And the tail portion, which are the non-polar portion, they face each other and they are hydrophobic interior of the membrane. That means it is saying that the head, they are attached to each other and they try to remain towards the hydrophilic side, and the tail that remain the just opposite sides of the head. Okay. Next part is that the primary physical forces for organizing the lipid bilayer are the hydrophobic interactions. Okay. So one force help actually to remain contact with each other, that is the hydrophobic interactions. Okay. And three classes of lipid molecules are there. One is the phospholipids, another is the glycolipids and the sterol. So here first is that phospholipids. What are the phospholipids? Generally these phospholipids have the four components. One is the alcohol, maybe glycerol or sphingosine, and then fatty acids, phosphate and alcohol attached phosphate. Okay. So there are the four components. So actually this is saying that alcohol, this alcohol may be glycerol or sphingosine, they attach to the phosphate. Or generally four components, what are that? Alcohol, that means maybe glycerol or sphingosine, and fatty acids and the phosphates. These are the main components, and the alcohol attached with the phosphate. Okay. Next, that is saying that the fatty acid components are hydrophobic. Okay. The fatty acid components are the hydrophobic, whereas the remainder of the molecule that has a hydrophilic. Okay. So fatty acid components, these are the hydrophobic in nature, and other portion of the molecule that are hydrophilic in nature. Okay. Two types of phospholipids are there. One is the glycerophospholipids and another is the sphingospholipids. And I have already explained about this structure. This is very rough structure. So after this, I will try to continue the remaining structural part of the plasma membrane. Okay. Okay. So phospholipids, that generally these are derived from the glycerol. Okay. And these are known as the glycerophospholipids, and I have given a short name, that is the GLP, for writing easily, that's why. Okay, so this is the phospholipids. These are derived from the glycerol and known as the glycerophospholipids. These glycerophospholipids that contain one glycerol, fatty acids, phosphate and an alcohol, and this alcohol may be choline. Okay. And according to the presence of alcohol, GLP or the glycerophospholipids, these are classified. For example, as alcohol is choline, then the molecule is called the phosphatidyl choline. Okay. The molecule is called the phosphatidyl choline. And if this alcohol is serine, then the molecule is called phosphatidyl serine. Clear? So according to the presence of alcohol, generally glycerophospholipids, these are named or classified. Next, these phospholipids are very numerous. Okay, and maximum number of these phospholipids are seen in the plasma membrane. Next one is the sphingospholipids. These sphingospholipids that contain amino alcohol, that is called the sphingosine. Okay, sphingosine, fatty acid, phosphate and alcohol attached to the phosphate. Here the amino group also present. Why the amino group is present? This amino group of the sphingosine backbone is linked to one fatty acid by an amide bond. Okay, that means the amino group of what? This sphingosine, amino group of this sphingosine, this is linked to one fatty acid by one amide bond. Okay, and this sphingomyelin is the most abundant sphingophospholipid. So here the glycerophospholipids, these are numerous, and obviously the sphingophospholipid also abundant here in the plasma membrane. And the plasma membrane of the animal cells that contains the phosphatidylserine, phosphatidylalanine, phosphatidylcholine and sphingomyelin. That means these 4 types of lipids generally seen in the animal cells. Okay. So after phospholipids, there is another lipid, that is the glycolipids. These glycolipids that contain the carbohydrate, may be monosaccharides or oligosaccharides, and covalently these attached, that means this carbohydrate covalently attached to the lipid, and these can derive from the glycerol. Okay, here generally these phospholipids also derive from the glycerol, and these glycolipids, these also derive from the glycerol or sphingosine. Okay, these derive only glycerol, but these can derive from glycerol or sphingosine. And the simplest glycolipid example is the cerebroside, and here only single sugar is present. Okay, and the sugar may be glucose or galactose, and the another complex lipid, glycolipid, that is the gangliosides, and here branched carbohydrate chain present here. And these glycolipids are found generally where the outer leaflets of the plasma membrane. Clear? These glycolipids are generally found in the outer leaflets of plasma membrane. Clear? So after that, another lipid present, that is the sterol, and I will explain in the next part. Okay? Okay, so now the sterols. What are the sterols? The basic structure of the sterol is the steroid nucleus. Okay, these are the steroid nucleus, and then that consisting of the 4 fused rings. Okay, 4 fused rings are there, and 3 of which that containing 6 carbons and 1 only containing the 5 carbons. Okay, and it is a planar. Relatively, I think so. 1 second, I have to check first. Okay, so it is a planar and relatively rigid structure. Okay, and if there is any sound from that coming from the outside, please adjust. And it is a planar, relatively rigid structure that is the sterol. Okay, first of all, what I'm saying, that this sterol is nothing but the steroid nucleus, and then these containing 4 fused rings, and 3 of which that containing 6 carbons and 1 with 5. Okay, and this is very rigid structure. And cholesterol is the major sterol. Okay, cholesterol is the major sterol that present in the animal cell, but in plant cell they lack the cholesterol. They haven't any cholesterol, but they contain another sterols like stigmasterol, cytosterol, and certain lipids also, that cholesterol and sphingolipids. Okay, and in the plant cells no cholesterol. That means in absence of cholesterol, there are another sterols, that are the stigmasterol and cytosterol. Okay, so here certain this is the structure of the sterols, or you can say the sterols, this type of sterol that is found in the lipid bilayer structure. Okay, so here now we will learn about that how the lipids are arranged or organized. Okay, though generally the lipids are organized in a form of wraps. Okay, in a form of wraps. These wraps are the membrane microdomains and that are enriched with cholesterol and glycosphingolipids. Okay, glycosphingo I think is necessary. 1 second. Glycosphingolipids. So the cholesterol and the glycosphingolipids. Clear? So what I am saying that the lipids are organized in a form of wraps, and these wraps are the microdomain, okay, and that containing cholesterol and glycosphingolipids. And these microdomains that also contain specific proteins, okay, and these wraps are generally two types. One is non-cellular and another is caveolin. Okay, caveolin means there one protein, that is caveolin protein, is present. Okay, and now another thing is saying here that when amphipathic lipids are generally mixed with water, then three types of aggregations or aggregates are formed. In the first case, that the fatty acid salt, which contains only one fatty acid chain, the molecules form a small and spherical micellar structure. Okay, spherical, just like this, spherical and micellar structure. Micellar structure. Clear? And these are here the hydrophobic fatty acid chains. These are hidden inside the micelle. Okay, hydrophobic, which are generally phobic to the water. So these are generally hidden inside the micelle. This is one arrangement is seen when lipids are mixed with water. In another arrangement, it is seen that a bilayer is formed. Here the two lipid monolayers that combine and to form a two-dimensional sheet-like structure. Okay. One here the two monolayer, that means like that two monolayers. Okay, these form a two-dimensional sheet-like structure, and in the third case, here a hollow sphere-like structure is formed by the lipid bilayer. Okay, like that hollow lipid bilayer structure is formed. Clear? And here this is called the liposome. And liposome is nothing but a closed lipid fluid containing vesicle, and that are bound by only a single bilayer. Okay, suppose this type of bilayer is seen here. Okay, this type of bilayer is seen here, and the fluids present within this hollow part. So three types of arrangements are seen when amphipathic lipids are mixed with water. Okay. And here one thing is very important, that is the asymmetry of the lipid bilayer. What is the asymmetry of the lipid bilayer? Here it is seen that the phospholipids are asymmetrically distributed across the bilayer, and the amine containing phospholipids are enriched in cytoplasmic surface. Okay, amine containing phospholipids that are enriched where? In the cytoplasmic surface. That means this is the cell, so this is the cytoplasmic area, this is nucleus. So cytoplasmic areas that containing the, what is that? The amine containing phospholipids, and the choline containing phospholipids and the sphingolipids, these are on the outer surface. That means here, these are the outer surface. So here, that means in the cytoplasmic area there are another lipids, and in the outer surface, that is there are the another lipids are seen. So this is the asymmetry of the lipid bilayer. Now we will learn about the what kind of proteins and carbohydrates that are present in the plasma membrane. Okay, so it is a vast video, so time is very important here, so I have to continue this briefly with brief structure. Okay. So now we will learn about the membrane proteins. What membrane proteins are there? Generally, two types of membrane proteins are there. One is the peripheral membrane proteins and another is the integral membrane proteins. Okay. So many proteins. First of all, we have to know what are the peripheral proteins. The proteins that associated with the membrane can released from the membrane by relatively gentle extraction procedure. Okay, sometimes proteins that released from the membrane, okay, some due to some extraction procedures. What are the extraction procedures? Like that, exposure to the solutions of very high or low ionic strength or of extreme pH. If any membrane that contacts with high or low ionic strength solution or extreme pH solution, then some proteins are released from the membrane. These are called the peripheral proteins. Okay, and here these procedures interfere with the weak non-covalent interactions between protein or protein membrane lipid. That means here what type of proteins are there? If protein structure is there or protein membrane lipid structures are there. Okay, but here lipid bilayer structure is intact. Okay, again I repeat, these procedures generally interfere with weak non-covalent interaction between protein or protein membrane lipid structure. That means this solution generally in a membrane, okay, in a membrane there are proteins, maybe suppose there are protein and lipids are there, okay, lipids are there, or sometimes proteins are there. Clear? So if the exposure is occurring, then it generally protein lipid structure or protein structure that is broken down. Clear? And these are the peripheral proteins. So either proteins released from the protein structure or released from the protein lipid structure. Okay, but leaving the lipid bilayer intact. That means lipid layer here is not hampered. Okay, only the proteins are released, and these are usually bound to the membrane, but these proteins loosely bound indirectly by interaction with the integral proteins or directly by interaction with the lipid polar head. Clear? So due to this exposure, when the proteins are released, these are loosely bound. That means not completely released from the membrane. If this is released, not attached with the membrane, then it is not attached with the peripheral proteins related to the plasma membrane. Clear? That means these proteins are released but loosely bound to the, not completely released. These are loosely bound with the plasma membrane by attaching with the integral proteins or attaching with the lipid polar head groups. Clear? So these are the peripheral proteins. These peripheral proteins are soluble in the aqueous solution. So here is the example of the peripheral protein, that is the spectrin, and it is found in the RBC. Okay. Next, the proteins that are held in the lipid bilayer tightly. That means some proteins that are not released or that are present very inside within the lipid bilayer, these are called the integral proteins. Okay, and these are transmembrane proteins also. These are known as the transmembrane protein also. Okay, which have one or more segments that ended in the phospholipid bilayer. That's why these are not released or try to release from the lipid bilayer, because they have one or more segments that totally attached or embedded within the phospholipid bilayer. Clear? These contain non-polar amino acid residues. These have non-polar amino acid residues with hydrophobic side chains. Okay? Hydrophobic side chains that interact with fatty acyl groups of membrane phospholipids. That's why these are not released completely. Why? Because they have non-polar amino acid residues and hydrophobic side chains that attached with the fatty acyl groups, clear, of the phospholipids. These transmembrane proteins are amphipathic, okay, and these may be single pass. Why the single pass? These are called the monotopic. That means single pass means only one ion can be passed through this, and multi-pass or the polytopic. That means two or more ions can be passed. And example is that the glycophorin in the RBC, that is the single pass protein, and band 3 protein in RBC, that is the multi-pass protein. Obviously, two are the examples of the RBC. These membrane proteins, which are covalently bound to the lipid molecules, called lipid linked membrane proteins, clear, which are bound to the lipid molecules known as the lipid linked membrane protein, okay, and they form attachment. That means this type of proteins attached with the lipid, generally three types of lipids are involved here for this attachment. One is the isoprenoid group, another is the fatty acyl group, and third one is the glycosylated group. The example of the isoprenoid group, that is the geranyl residue. Fatty acyl group example is the palmitoyl residue, and glycosylated example is the phospholipid. Okay, so these are the membrane proteins today we have learned here. Okay. Next, another is very important, that is the cell coat or the glycocalyx. These are the carbohydrates or carbohydrate chain. These are present on the surface of the eukaryotic cells, and these carbohydrates generally oligosaccharides and occur both as the glycoproteins and glycolipids. And this plasma membrane that contains also proteoglycans with surface exposed polysaccharide chain. Sometimes not only glycoproteins or glycolipids present, sometimes we can see the proteoglycans, and these proteoglycans have the side chains, and these chains are exposed polysaccharide chain, okay, and these are seen just upper surface, or you can see the just over of the lipid bilayer, okay, surface, just upper surface of the lipid bilayer, okay, this chain, proteoglycan chain. And the cell coat's function is what? That prevents the chemical damage of the plasma membrane, okay. So this is the very brief structure of the plasma membrane I have explained here. So you can consult any book after seeing this video. So this can be easily understood by you, okay. So thank you, and continue with my channel.